Okay, so I'm going to go over biological molecules with you, and I'm going to do the slideshow in 4 small chunks for those of you that need to squeeze this in around jobs, dinner, etc. So you can start watching them whenever you want to watch them and go through them. I'm hoping you can get each down to about 10 minutes. So the learning goals for the lesson: by the end, you should be able to identify and describe the 4 groups of biologically important molecules: carbohydrates, lipids, proteins, and nucleic acid. And this is going to be essential because we're going to need to have an understanding of lipids for this unit and proteins to a lesser extent in this unit. Carbohydrates will come up in this unit, but huge in Unit 2 when we start talking about metabolism. And nucleic acids, you need to understand nucleic acid structure and function for our unit on molecular genetics. So these are key concepts that underpin the whole course. You need to be able to identify the subunits that make up each macromolecule. So you'll know that you can do what you need to be able to do for the course successfully if you can describe the structure and function of the 4 major types of macromolecules. You can sketch them. You know the functional groups present in each type of macromolecule. So if you're still not comfortable with functional groups, take a look back over the functional group screencast, or see me. I can recommend some animations. We can sit down and do some extra practice, or you can always go find a Khan Academy or something similar. Will produce those if you need just a different explanation maybe than what I'm giving you. You should be able to sketch and name the biochemical reactions that produce each type of macromolecule, and you can compare and contrast the different macromolecules in terms of their structure and their function. So biologically important molecules tend to be called macromolecules because they're big. They tend to be polymers that are long chains made up of many smaller molecules linked together, with the exception of lipids. And lipids are big molecules in terms of the number of atoms, but they don't have that repeating structure that characterizes proteins, carbohydrates, and nucleotides. And the small molecules that make up a polymer is called a monomer. So poly, the prefix means many, and mono, the prefix means one. So it might be a good idea to start yourself kind of a diary or a terms section at the back of your notebook where you're kind of jotting down what some of those prefixes mean, because it will help you as we go through. So the first macromolecular group that we're going to look at are carbohydrates. And carbohydrates get their name because they're made up of carbon, hydrogen, and oxygen in a proportion of 1 to 2 to 1. So 1 to 1 carbohydrate, because this H2O is like water. They contain 2 functional groups, always: a carbonyl group, which could be either an aldehyde or a ketone, and a hydroxyl or an alcohol group, the COH. So they tend to be hydrophilic or polar and dissolve in water. So remember, hydro is water, philic is loving, so water loving, they dissolve in water. The opposite of that would be hydrophobic, phobic meaning fearing. So things that are hydrophobic don't dissolve in water. The roles of carbohydrates: they store energy, and it's quick and easily accessible energy. So we have carbohydrates in the form of glucose in our blood, and then carbohydrates as a polysaccharide stored in our liver. That's the polysaccharide glycogen. We also have, plants also store their energy as the polysaccharide starch. They should make up about 55% of your diet. And the monomer is referred to as a monosaccharide. And monosaccharides typically have between 3 and 7 carbon atoms in them. If a monosaccharide contains more than 5 atoms, they form a ring structure when they're dissolved in water. So what happens here is our cyclic glucose. You can see that here's carbon 1, and this is carbon 6. So the hydroxyl group can actually come around and interact, or sorry, the aldehyde group is going to be able to come around and interact with this hydroxyl group. And so the aldehyde becomes a hydroxyl group here, and this O is shared in the ring. So your rings will always have an oxygen in them. And then we've got another alcohol methyl group sort of projecting up the ring. We can classify monosaccharides in different ways. One way we can classify them is by the number of carbon atoms in them. And that classification you can see on the top here. So we can classify by number. A triose sugar has 3 carbons in it. A pentose sugar has 5 carbons in it. A hexose sugar has 6 carbons in it, and so on. We can also classify them based on whether they're aldehydes or ketones. So in this case, the aldose sugars and ketose sugars would be isomers of each other. They would have the exact same chemical formula, but their atoms are located differently spatially. So here, the aldose 3-carbon sugar is glyceraldehyde. That's going to be hugely important in the next unit. And dihydroxyacetone is the triose sugar that's the ketose sugar. All right, the C double bond O is in the middle. The C double bond O is at the end. And so they get slightly different names depending. And we can also look at the different types of carbohydrates in terms of monosaccharides connected. So 2 monosaccharides connected together is a disaccharide. Sucrose is a good example of a disaccharide. That's table sugar. Oligosaccharides are shorter chains of monosaccharides. So between 3 to 6, sometimes as many as 14 will still be considered an oligosaccharide. Polysaccharides are many monosaccharides joined together in the hundreds to thousands. So starch, glycogen. You can also classify from a more dietary perspective whether something's a simple carbohydrate or a complex carbohydrate. So simple carbohydrates are smaller sugars, not a lot of indigestible fibers. They're easy to digest. Complex carbohydrates are large and harder to digest, usually because they're more heavily branched and things like that. The bond that holds 2 carbohydrates together is a glycosidic linkage. And a glycosidic linkage occurs when 2 hydroxyls on 2 neighboring monosaccharides come together, and we call it a condensation or a dehydration reaction because, as you can see here, my 2 hydroxyl groups are going to react with each other, and water is what's going to leave. So if you're really stuck on a test and you can't remember what's going on at all, see if you can figure out a way to combine 2 functional groups to release water, because that's pretty much what's going to be produced in all of these. We can say the linkage is an alpha and or a beta linkage, depending on which way the hydroxyl group is pointing, and we define it based on which 2 carbons are involved. So you can see here we've got carbon 1 and carbon 4. So we call this a 1,4 glycosidic linkage, and this is an alpha linkage because both the hydroxyls are on the same side. If the hydroxyl groups were on opposite sides, that's what we call a beta linkage. So you can see that here, the alpha 1,4 linkage, they're both on the same side. Here's a beta 1,4 linkage. One is on the top and one is on the bottom. And that really just depends on which way that hydroxyl group goes when the ring structure forms. So I already gave you a few examples. These 3 are all structural isomers of each other. So they all have the molecular formula C6H12O6. And this becomes particularly important in biology, even more so than chemistry, because the biological properties of a structural isomer are quite different. You can't digest glucose using the same enzymes you use for fructose and vice versa. So glucose is blood sugar. That gets taken into your body and absorbed in the small intestine, and it's the sugar that body cells use first for energy. Fructose is found in fruits. It's also an easy transport in the blood and can be used by your cells. And then galactose is found in milk. So it's 1/2 of the disaccharide lactose. And each of these have 3 different 3D shapes, as I mentioned, which means the body's going to treat it quite differently. So fructose, for example, interacts with taste buds more, different taste buds or different receptors on taste buds. So it ends up tasting quite a bit sweeter than glucose does. Disaccharides, lactose, as I mentioned, maltose. Lactose is obviously the sugar found in milk and dairy products. Many people lack the ability to digest it. Pretty much every species but humans, every mammal, mammalian species but humans that do nurse their young, they tend to lose the enzyme around time for weaning. Humans, for some reason, have evolved a mutation so that they keep that enzyme into adulthood mostly. But obviously some people don't have that ability and they are lactose intolerant. Maltose is malt sugar. That's 2 alpha glucoses. And sucrose is a glucose and a fructose molecule, and that's table sugar. Examples of polysaccharides. We can have energy storing polysaccharides. We can also have structural polysaccharides that are usually indigestible. So starch and glycogen are the 2 energy storage ones. Glycogen is just basically the same as starch except more highly branched. And that allows it to be broken down more quickly because the enzymes that break it down can start attacking from all of the different branches as opposed to having to go from the end and work their way down. When you did your cracker experiment in our warm-up lab, what you were experiencing was the enzyme amylase in your mouth beginning to break down the starch in the cracker into glucose molecules. So if you kept it in your mouth long enough, you would have started to taste the sweetness of the glucose being produced. Cellulose is the common polysaccharide that's used to make plant cell walls. It's indigestible. Organisms that can digest cellulose have bacteria in their guts, which contain the enzymes necessary for its breakdown. And another structural polysaccharide is chitin, which is found in the exoskeletons of insects. I just want to make sure this is still recording. Yes, it is. Okay. I'm paranoid. Sorry. I will resume. These are some different examples. Idraparinux is a synthetic anticoagulant. That's an oligosaccharide. You can see it's got 5 rings joined together. It's also got some modifications on it. So it is an oligosaccharide, but it's an oligosaccharide that's substituted. There's an example of a ball and stick model of glucose, and this is showing you the starch in a plant cell and the cellulose fibers in the plant cell wall. And that's the end. I really should have waited. I apologize. And so you can stop and move on to